So we're going to start today by going over what I think are the most exciting developments in the field of single-cell genomics over the past year. I have 8 areas that I want to highlight today. They cover both computational and also experimental advances. And the goal is not to go into too much detail for any individual method, but I hope to really summarize the intuition behind each advance here, tell you why I'm excited about it, and maybe convince you to give it a try. And everything on this list is something that we think you should be aware of, even if it's only in preprint form. And in fact, we've made a particular effort to prioritize highlighting papers on bioRxiv because many of you may not have actually seen them yet, but they could be valuable for your work. So if you find these methods intriguing, we hope you'll check them out and read the paper. And also for each method, I'll try to give some historical perspective, on how they work and some of the older work that sort of paved the way for where we are now. And we will make a video of this talk available after the workshop. So I hope that will be helpful. All right, so the first method I'll tell you about is an experimental trick, but it's a good one, and it can really cut your costs. And to start, I'll just remind you how droplet-based assays from companies like 10x Genomics work. They place single cells into droplets and then add a barcode. Very straightforward. A downside, though, as many of you know, is that most of the droplets that are generated don't actually have any cells in them. And you can already see that in the schematic. Like this droplet here, it has a cell, but these two, for example, do not. And actually, in a real experiment, more than 90% of the droplets that you generate are empty. Now, that's not the end of the world, but it is kind of a waste. You're paying, 10x good money for those droplets, and then for these empty ones, you're not actually getting anything back. And so people have been trying to get around this problem for a while, and the goal or the idea is pretty simple. Instead of leaving some droplets empty, why don't we fill all the droplets? In fact, let's put multiple cells into each droplet. And if we give them a combination of barcodes, then you can tell them uniquely apart. So that idea was first put forward by Paul Dattlinger and Christoph Bach in a method called SciFi-seq. And the idea is that you first put on a barcode into cells in a 96-well plate. So they already have a barcode even before you load them into a 10x machine. Now you can put 4 or 5 cells in a droplet. There's no more empty ones. The 10x machine adds a second barcode, and you can use the combination to tell the cells apart. So the good news here is you get a massive increase in throughput. Instead of about 5,000 to 10,000 cells per experiment, now you can get 150,000 cells out of a single channel. But the downside is that the data quality is a little low for this method, even when just working with cell lines, as I'm showing you here. But this year, a couple of groups figured out how to overcome this. And the trick was just to switch the order of barcoding. So the first method is called UDA-seq from Westlake University. And the first step is very similar to a normal 10x run. You just add a lot more cells into the instrument so that multiple cells, like I'm showing here, go into each droplet. And that process adds your first barcode. But the wild thing is, in that UDA-seq, the cells actually stay intact throughout this process. They don't lyse, even when you break the droplets. So now, in a second step, you can put cells into a wells of a 96-well plate and add a second barcode with the pipette. And it turns out that this strategy is a much better molecular way to accomplish this task. Do the 10x barcoding first, and then you add your own barcode in a 96-well plate. Do that second. So there are real advantages to this approach. The first is that it maintains this huge increase in throughput. So you still get about 150,000 cells per channel, and that probably drops the cost of 10x library prep by almost an order of magnitude, if not more. So that you're really saving kind of thousands of dollars with this trick. The second advantage is that this approach is really compatible with diverse assays, even multimodal assays, not just RNA-seq, not just ATAC-seq, but also the 10x multiome, which is traditionally a very expensive assay, and this is a great way to cut costs. And you get good data quality. And most importantly, it doesn't just work in cell lines. So these papers show success in multiple tissues, including human blood, the human retina, the human kidney. So this is very exciting data. So I think it's a nice trick. I think it's a really good way to cut the cost of commercial droplet-based assays. I know 10x is looking into this. If you're interested, you should too. And if you're interested, Spiros Darmanis, whose lab at Genentech developed one of these methods, will be telling us about it this afternoon. All right, so next I'm going to talk about some computational tools to make sense of all of this data. So those of you that have followed this field for a while now know that reference mapping techniques are becoming very widespread. As we recently wrote in the perspective, reference mapping follows the trend of human genomics. Whenever we do an RNA-seq experiment, we map the reads to a reference. We leverage the incredible amount of work that the Human Genome Project did in order to make our lives easier going forward. So we want to do the same thing for single cells. When we generate new data, we don't want to go through it and manually cluster it each time. We want to leverage existing prior work to help us interpret this new data, ideally in an automated way. And that's what reference mapping aims to do. So there's a variety of tools that do this now, and they tend to fall into two categories. The first are tissue-specific references. Those are centered around individual organs like the brain or the blood. And these are often trained by domain experts with tissue-specific knowledge. But another route is to look across the entire body. And that's what foundation models like SCGPT or Similarity or others are trying to do. They aren't limited to any one tissue. They aim to analyze cells from any human experiment. And that's a really exciting development. And these models are really cutting edge, especially as they're often trained on manual annotations from large resources like CZI. But that does create a problem, and those of you that have used these methods know this. These models are trained on a lot of data, but the data are annotated by different groups using different ontologies and at different levels of resolution. So here's an example for the heart. You can see that the data is good, and there's a lot of it, but the labels are a mess. And so our take on this is it's not just about the neural network, it's not just about the amount of data, it's about having well-curated data that is consistently, accurately, and uniformly labeled. And my lab has spent a couple of years on this problem. What we've been doing is not just gathering a lot more data, but going through one tissue at a time and trying to annotate cells with a consistent hierarchical cell ontology that is uniform across tissues. We've been doing this one tissue at a time. We have 23 of them now. We've been building up a large corpus of training data. And once we build that training data resource, then we feed it into a neural network. And we call that neural network pan-human azimuth. And pan-human azimuth can now classify cells uniformly and robustly across the entire human body, regardless of the specific tissue and regardless of the technology. And what I think makes this special, it's not just the architecture of the neural network or the amount of the training data, it's the effort we put into curating and QCing the labels. And so I hope what's exciting here is how powerful this is to annotate new data. So here we're using it to manually annotate, to automatically annotate and interpret the entire Tabula sapiens V2 reference from CZI. That reference spans the entire human body, but we can process and annotate it all in one single parameter-free command. Now, for the results here, there are hundreds of labels. I obviously can't show you them all, but we think they agree with and very nicely augment the manual annotations from the CZI team. You'll see more of this later today. But we think this is exciting, and it's also very highly scalable. So we've actually mapped the entire SC Basecamp repository of human cells from the ARC Institute. That's something like 85 million cells. Took us less than a day, again, with the same uniform set of commands. And I know that these UMaps don't really tell you very much on their own. That's also why we make heat maps to allow you to check the quality of these annotations and confirm that they match expected expression patterns. We make these every time we annotate and encourage you to do so also. So we're really delighted to openly release Pan-Human Azimuth today. You can try it out for yourself. cetjalab.org/Pan-Human Azimuth. It's free, it's open source. You can try it if you're an R user, if you're a Python user. It works with Seurat, it works with ScamPy. Really easy to install and run. And if you want to hear more about it, Skylar and Saurav, who are the primary authors, they are our next speakers after me today. All right, so let's transition now to spatial technologies. And many of you are aware of rapid developments in this space with technologies like SlideSeq and slide tags. And these methods work by having a collection of spatially barcoded beads that are randomly packed together into what's called a puck. So what you do is you lay your tissue on top of the puck, the barcodes mix with the tissue, and you get beautiful spatial data. And this really works, it's a fantastic approach, and it works nicely across many tissues. However, one thing which is a bit tricky is that since the beads on this puck are packed randomly, you have to figure out where they are. You have to figure out the spatial location of each barcode. And you accomplish this with a procedure called in situ puck sequencing, but it is a relatively involved process. It involves a lot of microscopy and a lot of sequencing by ligation. So it takes time, it takes money, and it can be a limiting step in this process. So we'd love to get rid of it. Basically, we'd love to be able to generate this nice spatial data on the left without going through this laborious process on the right. But how can you learn the spatial location of a barcode without doing any imaging? And that's the advance for this year. And it really leverages a cool mathematical trick. So let's say that you wanted to draw a map of all the cities in the US, but you didn't have a satellite. But what you do have is a list of all the cities and the pairwise distances between them. And it turns out that even if this is all you have, just knowing who is next to who, you can reconstruct an entire physical map of the United States. You can infer location from pairwise distances. That's a really cool trick, and you can do the same thing with spatial analysis. So the strategy here, also from Evan and Faye, is to figure out the pairwise distances of beads on the array. And they do that by introducing something called diffusible beads. I don't have time to explain exactly how they work, but here's a simple analogy. Let's say you're on the phone with someone, and right next to you, a car sounds its horn. If you can hear that car horn in your own ears, but also through the phone, then you know that your friend must be close by. So that's basically what's happening. You have thousands and thousands of these diffusing barcodes acting as random car horns. And if the car horns are really loud, then you know two beads are very close together. And that information alone, even without doing any imaging, is sufficient to computationally reconstruct the location of each bead. It's pretty crazy, but it really does work. It works in simulated data on these pucks here reconstructing the letter H, but most importantly, it works on real data as well. These two data sets below show real spatial data, either on the mouse brain or on an entire P1 head that was collected entirely without imaging. I think this is really exciting. It means you don't need a custom imaging setup. It means you can do much larger spatial studies. And I think what this starts to point towards, and this is a vision that I heard Faye present, is it makes the spatial part of data collection so easy that even if you just want single-cell RNA-seq, you'll do the spatial anyway. Why not? The information basically comes for free. And I think that when that happens is when you're really going to start to see the single cell and spatial fields come together. So I think this is a big deal. If you want to learn more, we're excited to have Faye and Evan's graduate student presenting later today. And I also want to acknowledge a number of other groups who have been pushing similar visions. All right, so I'm going to transition now to like another sort of crazy experimental idea that was very recently put on bioRxiv, really only about a month ago. Again, it has to do with droplets. Droplets are a very powerful approach to be able to capture cells and reagents together. They're like little reaction chambers, right? But one downside with droplets is that once you make them, whatever is inside them when they form, you're stuck with that. You can't put new reagents in, you can't take them out. The droplets really are sealed. But this year, multiple groups, including Alon Klein's, figured out an incredibly cool new trick, and they introduced something called cages, or capsule with gel envelope. There's some really cool physics and chemistry that use a polymer called F127DA. Definitely can't fully explain it, but the point is that you create a robust polymer shell that can encapsulate cells and reagents. So if you zoom out, it looks just like a droplet, like in this picture here. But if you zoom in, you can see that it also has pores. Like you can see in this electron micrograph, those pores are 10 to 50 nanometers. So you can add things into the cage and you can subtract them out. You can add enzymes, you can add short DNA primers, you can even add media, but bigger things like cDNA, like long nucleic acids, those stay inside the cage. So you can do multi-step reactions and washes, but not lose the cells and molecules that you care about. So this is pretty cool, and there's a wide range of applications here. So for one, you can start to do complex molecular protocols on individual cells, like combinatorial barcoding protocols, for example. You can successfully add barcodes to cells, and the molecules over multiple different steps, the cells and the cDNA, they happily stay in their cages, isolated from one another throughout the entire process. So here, the authors introduce their own version of single-cell RNA-seq, for example. This proceeds with this combinatorial barcoding process. The experiment performs well, and they actually show that their data actually outperforms the 10x platform in this case. But what's really cool here is that you can add media so the cells can actually grow inside the cages. So I'm showing you that here. When you start, you only have one cell in a cage, but then over time, you actually grow these little colonies. You can expose these different cages to drugs, and then you can do molecular assays like RNA-seq to see how each colony or even how each cell responds. So I'm not showing a lot of biology here because the preprint is very new, but I think conceptually, this ability to grow cells, to manipulate them, to perturb them, to barcode them, all these little cages, I think it's pretty cool. I think there's going to be a lot of biology that comes out of this vision that the Klein Lab and others are just working out. So there are a few groups working on this. They all have bioarchive preprints. You can check this out. A Lux portfolio company called Atrandi Biosciences is also commercializing this approach. So please feel free to read more. All right, so the next method that I want to show you today is focused computationally. And the idea behind it, which is a bit funny to say today, is that we're too focused on our cells. And let me tell you why I say that. When we do single-cell RNA-seq, we think about labeling individual cells and cell types. So we look for changes in cell state. Maybe we'll look for differential expression between cell states or cell types. But really, we're focused on our cells, one at a time. But what I think a number of groups are starting to realize is that sometimes when you're doing single-cell analysis, especially when you're doing case control analysis and comparing two groups of samples, we don't primarily care about how to describe or label any one individual cell. What we care about is how to analyze a sample, how to analyze an entire distribution of cells that span a range of types and states. So my goal here is really to introduce you to this challenge, which I think is critical to the field. How do we change our mindset? How do we go beyond analyzing individual cells one at a time and move towards analyzing samples? And if you're new to this field, or even if you're not, I think that this problem of developing methods to assess sample level heterogeneity is one of the most exciting and important areas in our field right now. Let me show you why this matters. So here are three analyses that are very common for single-cell RNA-seq. How do I cluster my cells? How do I build trajectories? How do I map information from reference to query? There are dozens, if not hundreds, of papers on each of these problems at the cellular level, but I want to draw an analogy for samples. So for clustering in a single-cell RNA-seq experiment, clusters of individual cells correspond to cell types and states. But if we could cluster samples based on their distribution of cells, then those sample clusters could correspond to different patient groups. They could correspond to different patient cohorts. Maybe they have different risk profiles. Maybe they have different treatment outcomes. Those are important things to discover. Similarly, for trajectory inference, pseudotime for a cell models a cell's individual transition, but a sample level trajectory could represent the timing of disease within a patient, the disease severity within a patient. It could allow us to understand how coordinated changes across cell types drive disease progression. Lastly, we say that reference mapping can often be used to annotate the state or type of a single cell, but what if we could do this for a sample or for a patient? What if we could look at someone's blood and based on the distribution of cells that they have, we could say, I think that person has severe disease, or I think that person is likely to respond to therapy. That's what we're really interested in. So we need new methods for sample level analysis. I'm not the first person to say this. I certainly can't cover every new approach that's thinking about this angle, but I want to bring attention to this problem and to this field. All right, so first, I should want to say there's a series of generative models that can be developed and tailored to address this problem. So one example is SC-Poly from Fabian Theis's lab. This is a variational autoencoder framework. It's very popular for single-cell data integration, taking cells from different studies and integrating them together. But it has some cool modifications that enable us to do PCA not at the cell level, but at the sample level. So each dot here is a sample, a different individual. And now you can see how different individuals group together based on their cell distribution. And you can see these little clusters of samples emerge, and those clusters form for two reasons. One is a batch effect from study to study, but the other is that you can actually see separation of samples from organ donors and samples from live biopsies. They have different distributions, so that's cool to see. There are a variety of tools that use generative modeling to address this problem, including Mr. VI from Nir Yosef's lab. But another method that has been very impactful from my lab is SE-LKME. This is from the Yi lab at UNC. And what it does is it takes a single-cell RNA-seq sample and says, okay, we're not going to worry too much about any one individual cell. What we do care about is the distribution of cells together, a holistic look at the sample. And through some clever mathematical tricks, they can focus on the density of these cellular distributions and quantify that in order to understand sample-level heterogeneity. So they have this very cool example of a CyTOF data set from the cord blood of about 40 different newborns, but some of the newborns were born early, some of them were born late, some of them were born at term. So what the method does is it learns a trajectory at the sample level. So each dot here is a different newborn, and you can almost see a trajectory of gestational progression. It's like pseudo time or pseudo age, but for the sample. And in fact, there is quite a strong relationship between the pseudotime and the actual gestational age of the newborn. So this opens the door to asking, how does the density of cell states in a newborn's immune system change with age? Not just for one cell or one cell type, but for the entire system as a whole. These are really fun questions to look at. So if you're interested in these questions, please stay tuned for Long Dejiang's talk this afternoon. He will present a new method that my lab is developing that we're very excited about, and I hope will be very useful to many of you, especially if you analyze data from diseased patients. All right, so let's jump to another creative idea. The goal here itself is not new. It's an idea that's been promoted by many groups, but quite prominently, for example, by Udi Shapiro at the Weizmann Institute. So for a long time, for more than 20 years now, Udi has been arguing that the single-cell sequencing revolution was a unique opportunity not just to learn cell types, but also to build out an entire organismal lineage tree from the fertilized egg stage. And Udi basically singularly devoted his biological lab to this vision. So obviously, an organismal lineage tree would be an incredible accomplishment, not only for developmental biology, but also in cancer to understand clonal evolution. Speaking of lineage, I heard a lot about this project while I was a postdoc in Aviv Regev's lab because Udi Shapiro was her PhD supervisor. But anyway, obviously, this problem of building lineage trees is very challenging because how do we know what lineage a cell belongs to? The DNA mutation rate across cell divisions is very low, so it's hard to learn mutations and lineage from DNA data. And also, there are very few examples of ground truth to be able to develop and test methods on. So the field has primarily taken two approaches. The first is to integrate a clonal barcode into the genome using a lentivirus, for example, a cell tag. That works really well, but you can't obviously infect live human beings with lentivirus, so this requires model systems. The second strategy is to use mitochondrial DNA mutations. They have a much higher mutation rate, and they can be applied directly to human tissues. They are native lineage marks, but the resolution of lineage tracing here may be quite limited, especially in non-cancer samples. So the field is realizing that neither of these approaches can robustly perform lineage tracing in primary human tissues, so we need new methods. And there's been some really nice progress in this idea this year. And it builds upon an idea that multiple groups, including Dan Landau's at the New York Genome Center, have been talking about for a while. And the idea is to use epimutations, or changes in the epigenetic state of cells, in order to build these lineages. So as you know, DNA cytosine bases can acquire a methylation mark that is heritable over cell divisions. But cells can switch during a division in their methylation state. They can flip from on and off or vice versa. And that switch in methylation status, or an epimutation, occurs at about a 0.1% rate per cell division. So the idea is, let's use those epimutations as information for lineage tracing. If two cells have almost exactly the same DNA methylation or epimutation profiles, then they're probably part of the same clone, and they might even be sister cells. Now, by the way, some methylation sites are very strongly associated with particular cell types or states. We don't actually want to worry about those. Other methylation sites are just sort of random. Those random ones, they are neutral sites, and their methylation status therefore serves as a heritable lineage mark. So this strategy of building lineages based on epimutations, it really does work. This is a paper from Westlake University published this year. The authors did a very simple benchmark experiment where they took hex cells, repeatedly grew them and split them into wells in a way that they knew that the true lineage tree of these cells, and then they profiled DNA methylation status. And they built a computational approach called methyltree. And you can see here that you get a really remarkable agreement between the clustering of the DNA methylation profiles and the true ground truth lineage states. And it really is quite remarkable, especially as there was absolutely no experimental perturbation performed on these cells. Complementary work from Lars Velton's group also pursues a similar approach. In this example, they applied their technology to mammalian hematopoietic stem cells. So again, they profiled DNA methylation at single cell resolution. Now, if you cluster on all DNA CpG sites, the cells cluster as you'd expect by cell state, so you can see different populations of lineage-committed progenitors. But if you cluster only on the neutral sites, you start to see these little groups kind of pop up. And the hypothesis is that these groups are different clones and different lineages or sublineages that were present in the experiment. And what's really remarkable here is that in the same experiment, they also performed viral lineage barcoding with the Larry system. So again, they have ground truth estimates of clonality. And you can see there's a nearly perfect overlap between the clonal information derived from DNA methylation, which didn't require any experimental perturbation, and the ground truth lineage information. So I think between these two papers, there really is starting to be convincing evidence that DNA methylation epimutations can be used as heritable marks to assess clonality and lineage, even in primary healthy human tissues. I don't have time to show you biological results. You can read these papers. They explore a variety of different biological questions, including the degrees of oligoclonality in human aging, trying to determine stochastic fate choices in the early mouse embryo. And I'm sure there'll be many, many more types of these questions. We're excited to have Michael Scherer, who developed one of these techniques, who's going to present on this work later in our workshop today. All right, so my next topic is less about a particular method and more about sort of a unifying vision for the field of systems biology, especially as it relates to single-cell analysis. And the idea is to use artificial intelligence to build a virtual cell, a cell whose past, present, and future behavior can be simulated and predicted entirely in a computer. That idea has really sort of swept through the field this year. It's not necessarily a new goal in systems biology. The idea of modeling cells as circuits has been around for a while. But what's different now is that we have much better ways of generating data with single-cell technology, and we have much better ways of analyzing it with AI. So what is a virtual cell? Like, what does that even mean? There's not a single definition. There are lots of methods that are being developed towards this goal. But one thing that these methods have in common is that they want to be able to do a virtual experiment and predict its outcome. They want to be able to expose a cell to a drug or a genetic perturbation and predict what will happen, to be able to do a wet lab experiment in the computer. Now, the goal may not be to replace experiments entirely, at least in the short term, but it is to help design them more effectively. So for example, let's say that you have a library of 50,000 drugs, and you can't experimentally test all of them. But what you could do is you could do a virtual screen first, take the top 1,000 or so predicted hits, and just test those experimentally. So that's the idea. Virtual cells enable virtual screens. They can narrow the search space. They can help you prioritize your experiments. So I'll show you an example. This is a paper from Fabian Theis's lab. It describes a method called Prophet. It's a very exciting and ambitious paper. It uses a transformer architecture. It takes in all sorts of single cell and perturbation data, and it aims to scalably and universally predict cellular phenotypes. It aims to predict cellular viability, cellular gene expression, and even cellular morphology in response to perturbation. So as an example, they do a virtual screen of EGFR inhibitors in a large number of lung cancer cell lines. Now, some of these cancer cell lines are known to be sensitive to EGFR inhibition, but not all of them have been tested. Now, Prophet can screen all of these. It can predict a gradient of sensitivity to EGFR inhibition, and you can compare those results to the actual ground truth. Now, in general, these are the types of predictions that need to be validated in high throughput, but I think the goal and general idea of virtual cells is to use these types of predictions at least as a first good initial guess. Again, you can use that to prioritize particular cell lines for experimental follow-up, drug development, and treatment. So that's the idea. And it's in its early days, but I think one of the key assumptions is that even if these models and methods are a first step, they will benefit enormously from the explosion of experimental data that's being generated. Better predictions will lead to better experiments. They will lead to better data and even better iterations of these models. So I think it is sort of a virtuous cycle for virtual cells. It's a really fascinating hypothesis that I think multiple groups will be focusing on and testing over the next few years, if not the next few decades. So to learn more about this, tune in for our first keynote speaker, Yuri Leskovich from Stanford. He's really one of the conceptual thought leaders in this space, and he's going to tell us about the future of biology. It appears to be agentic. And I'm very excited for this. He'll be the last speaker in this session today. You'll also hear from Isabella Grabsky, who's a postdoc in my group. She's developed a new technique that we think will be quite valuable for utilizing perturbation data. It doesn't really create a virtual cell, but it will help us map and integrate perturbation responses across different experiments in biological contexts. We think this will be very useful to many of you, so please stay tuned for her talk this afternoon. All right, for our very last highlight of the day, I want to discuss what's I think is a major new opportunity for the field. It's been challenging for a while. It's the ability to infer transcription factor binding and transcription factor dynamics in single cells. This has been an extremely challenging problem traditionally because transcription factor DNA interactions are weak, they're transient, and they're really, really tough to measure. But this year, a couple of groups have figured out some tricks. And the first is Jason Buenrostro's group. Jason's one of the inventors of single-cell attack-seq originally. Most of us who do single-cell attack-seq, we focus kind of just on peaks of open chromatin. But just because a chromatin peak is open doesn't necessarily mean a transcription factor is bound inside of it. But here's the trick. If you look really closely at single-cell attack-seq data, if it is bound by a transcription factor, there is a dip in accessibility right at the site of binding. It's called a footprint. You can see it here in these cartoons, but it is in real data too. So these are known as footprinting analyses. You can see here that in some cell types, for example, there is open chromatin around GATA1 sites, but right at the GATA1 motif, right at the site of binding, there's a strong dip in accessibility. And that suggests this footprint that GATA1 is actively binding these peaks. So that's real evidence of binding. It's very cool. The challenge, though, is that this data is very, very noisy. These plots are generated by averaging the signal over thousands of peaks to get this type of visualization. So how can we clean this data up so we can get much higher levels of resolution? And this is where the advances come from. There's a really neat advance from Jason Buenrostro's group. It uses some really innovative computational methodology. I'll describe the intuition in two key steps. The first is to really remove the TN5 insertion bias that's present in single-cell attack-seq data. They build a cutting-edge deep learning model of TN5 bias with sort of best-in-class training data. It is kind of a somewhat technical bias correction neural network, but it's a huge resource to the field, and it really cleans up attack-seq data. Second, they use the attack-seq data and the DNA sequence together. If there's a hint of a footprint and it happens right over a transcription factor sequence motif, then those two pieces of evidence together are good evidence of binding. So I can't go into the computation in detail. Please do read the paper if you're interested, but I'll show you a couple of results. So this is an example of looking at footprinting data at the hemoglobin locus. So here we're looking at human bone marrow. They generated a variety of multimodal data and then analyzed the attack-seq data with their new methods. And you can look at the differentiation of erythroid cells. So at the beginning of erythroid differentiation, the hemoglobin locus, as you'd expect, is pretty closed off. They're kind of nucleosomes all over it. But as differentiation proceeds, at some point, a transcription factor binds and kicks off this nucleus at the promoter. That's step one. Okay, so you start to open up this locus. But as the differentiation continues, this footprint, which you can see here, it starts to get wider and wider and wider. And that's because additional factors can now come in and bind now that this locus is already open. So it's pretty cool to be able to look at these dynamics and trace them over differentiation, especially for such an important gene locus. Okay, but how do we actually know what factors are binding? So the method tells you that too. This is data that was generated in cell lines. Looking at just one locus, you can detect 5 different footprints. So in each case, the deep learning model predicts that the footprint corresponds to the binding of a specific and different transcription factor. And what's really remarkable here is that in every one of these cases, the predicted binding lines up precisely with a separate experimentally derived ChIP-seq assay of transcription factor binding that serves as ground truth. So again, I know I'm going pretty fast here, but please check out this paper from Jason's group to learn more. I think it's pretty exciting. I also want to show you an experimental strategy developed by Ivan Raimondi and Dan Landau's lab, and it's called D&D-seq for docking and deamination. So remember I said that transcription factor binding is hard to measure because it's transient and weak. So Ivan realized to get around this, you need to leave a permanent mark in the genome just to note that binding has occurred. And so what DNDC does is it uses a secondary nanobody fusion to recruit a very potent base editor to locations of the genome where a transcription factor is bound. Even if that binding only takes place for a short period of time, the base editor introduces a permanent CDU mutation in the genome, and then we can read out those edits in a single cell ATAC-seq assay. So it's pretty cool. The workflow is you stain your cells with an antibody against your transcription factor of interest and your DND fusion protein. You wait for a while, the mutations occur, then you run single cell ATAC-seq and you look for those mutations in your data. And when you see mutations, you can call transcription factor binding peaks. And so in this particular location, you can see they line up, in this particular case, you can see they line up nicely with separate encode ChIP-seq data for GATA1 and CTCF in K562 cells. The downside is this is pretty sparse data. You're measuring less than 100 binding events per cell. So you do need to sequence a lot of cells, and you need to pseudobulk them together per cell type. But as costs drop, even with things like UDA-seq, like I showed you this morning, very, very large-scale experiments are becoming more and more possible. And so I do think that these methods, both computationally and experimentally, will bring us closer to this really exciting goal of being able to explore transcription factor binding dynamics at single-cell resolution. All right, so that wraps up my overview of recent advances this year. It's always a roller coaster for me to give this talk and to acknowledge and celebrate the amazing work from this community over the past year. I hope you learned something and maybe found a new method or preprint that you want to check out and possibly try in your own work. Of course, if you're interested in learning more, please stay tuned for our wonderful and exciting speaker list today. If you have questions, please feel free to post them in the YouTube chat for speakers. Thanks very much.